Hello, my name's Deborah Phippard and I'm the head of research for Precision for Medicine. So Precision for Medicine is an innovative company that delivers true translational solutions for clinical oncology studies.
Hello, I'm Jerry Messerschmidt. I'm the Chief Medical Officer at Precision for Medicine. And as Deb said, we help small companies think through how to get their products to market efficiently to help patients.
So the introduction of biomarkers into clinical medicine is truly innovative and allows us to get drugs into the right patients faster. I think it's truly making a difference in how we do medicine. I've been working in this field for more than 20 years, and I truly don't want to run a clinical study unless I've got a biomarker translational program. Being able to stratify patients, make sure that we've got the right patient for the drug, that we know our drug is on target, that we're tracking our mechanism of action, that we're looking for outcomes, we're really understanding what's going on in a very complicated disease. You truly can't do that without an integrated biomarker program. And that's really what Precision brings to the clinical research piece. We've built extensive global capabilities, and we can bring many different assays to bear on how we think about the research. So the first step for us is always, okay, what's the drug? What's the mechanism of action? What's been done in the past? Do the companies we work with, do they have an idea of what they're looking for? Or is this something where we've really got to bring our scientists to bear and de novo think about how would we test for effects of this drug in vivo in a very robust way? And it sounds super simple, we're going to do a biomarker program, but I can tell you in reality, something that works at the scale of the research bench is truly not ready to go into the clinic. It's not robust enough, it's not reproducible enough. Maybe you've got transit problems, you need to get it to a lab that's not right next door to the clinic. You know, how do you think about putting all that together? And that's really where Precision brings many different disciplines together, everything from cold chain logistics to highly complex assays that we integrate with the clinical team and make it all work.
Biomarkers have been essential in oncology for a long period of time, but the numbers that we've been developing over the last few years have dramatically increased. And now we have, since the Human Genome Project, we have an understanding of the process from the gene to the messenger RNA to the protein. And these proteins, messenger RNAs, other RNAs, and even circulating DNAs and cells are really important for us to understand the biology of the cancer. And what we found is something that's not shocking to most people today, but that individuals differ in how their cancer actually grows and progresses within them, and understanding those differences within the individual is really important for making decisions for next steps in the development of drugs and for bringing the drug to market.
So I'd just like to pick up on one of Jerry's points, because I think he raises something that is so fundamental to cancer. And I know we're talking to a very educated audience, but you tend to think cancer is cancer. You've got head and neck cancer, but there are so many different molecular pathways that are activated in cancer. Cancer is a clonal, it's not one cell type, and you have to think very carefully. If you put a needle into a cancer, you take a biopsy, you're looking at one very small piece of that cancer. We know it grows, it mutates, so we're bringing newer technologies such as circulating tumor cells, where we're looking at, for instance, cells in the periphery. So perhaps these are the more dangerous cells. They're the ones that are in the circulation. We can look at how the phenotype is changing, how those cells become metastatic, more likely to lodge into different tissues. So there's lots of ways of thinking about how to explore the cancer, how to look at what's changing throughout the course of drug treatment, how we might want to add other modalities into that particular treatment program, and really understand in real time how a particular patient is responding to a drug and how we want to adapt our clinical studies to that. So we really think about adaptive clinical designs response. We think about what's the most likely tumor type to respond. We think about stratifying the patients. And you really can't do that with solid biomarker assays that you can run in real time, where you trust the data, because you're using that data to inform patient care. So you really need to think about, are these assays robust? Are they run in a good, well-regulated lab? And that's really what Precision for Medicine does. So I really would like to tell you about the benefits of integrating biomarkers into a clinical study. You can probably tell this is something I'm really passionate about. I'm a translational scientist. I always want to understand, when a drug is given to a patient, what exactly is that drug doing to a given patient? So humans are very heterogeneous. Unfortunately for us, we do our animal studies, mice are inbred, they all behave a certain way. We know when we get into the clinic, that's not going to be the case. So if you really want to understand who's responding and why, you need a very robust biomarker program to go alongside your drug. Heaven forbid you miss your clinical endpoint, but should that happen, if you've designed your biomarker program well, you will be able to go back, run the relevant assays, look at response, and perhaps you can see a subset of your patients are responding. Then you can understand what is different about those patients and use that to inform the next round of clinical testing. The old days, you have a failed clinical study, the drug is dead, might be a perfectly good drug. I mean, we're the victims of, did we design this trial correctly? Did we test things properly? If you put a biomarker program together with how you think about your clinical design, it allows you, if you like, to almost get a second chance. It allows you to refine, and we like to do that in the context of the clinical study, which is the whole concept of adaptive clinical trials, understand who's responding and why. So I really want to see a biomarker program go together with every clinical study when that's what we can do.
So biomarkers are extremely important, but not just as a group, but individually. And as Debs has mentioned, we help our scientists that are starting these new startup companies who are doing fantastic cutting-edge research, but may not have the experience to really know how to get it from the lab bench to the patient and then to approval. And that's where we look to help our clients figure that out, so that they can understand how their cutting edge research is actually translating outside of the research setting and into humans. And there are quite a few differences. Cancers are not cells that have brains. They are bags of chemistry, basically, biochemistry, and they do what they're told to do, basically by their DNA, their RNA, their proteins, but also things from the outside affect these. And when they get damaged, they can cause a cancer. And a cancer really is a very simple idea, is that it's a cell that never stops growing. And so it grows and grows and grows. And frankly, it kills humans by becoming too big in places like a liver or a brain or a kidney, essential organs that are required to function normally but can't because they've got this mass growing in them. But what else is occurring is actually much deeper and much more important, is the whole human system has to be taken into consideration. We spent actually 5-6 decades treating cancer to try to stop them from growing. But what we've learned is that we can stop some of them from growing, but these cancers have other survival mechanisms that have to be taken into consideration. For example, they mutate quite rapidly. In a paper published in Nature in 2012, an average of 7,000 tumors that were sequenced had over 700 mutations in each cancer. That's a lot of mutations. Many of those may not be actually functional, but that means a lot of those can provide survival benefits. 30, 40, 100 survival benefits over time accumulate. And the immune system plays a really critical role between the cancers and whether they survive or not. In fact, probably the immune system is really the only system that can actually kill every last single cancer cell. But what happens is that these cells mutate so rapidly over periods of time, become resistant to therapies, continue to have survival benefits. And that's what the biomarkers are trying to understand. What's going on inside the cells, inside those cancer cells, and what is being affected by the drug therapy and what is not being affected, what may be associated with toxicities in normal cells, and what may be happening with the immune system. And so today, the analysis of a clinical trial is much more complex than it was 10 years ago, in that we have so many more ways of understanding whether the drug is working. Is it working in just a specific population? Is it working in 2 or 3 populations, or not working at all the way that we thought that it might be working? And so biomarkers are the critical link between what's happening in our human patients and the knowledge that we gain from each patient to be able to make judgments for future treatments.
So I think about biomarkers, and I think about the old paradigm of drug treatment where you had to give a drug to a patient and then you wait to see if they respond. And I think everybody listening probably knows that every drug has a side effect. So there's always a downside to taking a drug, and who wants to take a drug if you're not going to respond to it? So to me, being able to take a patient, look at that particular disease and predict what of the many drugs we have in our toolbox is going to be best for that patient is incredibly powerful to me. So drugs are expensive, but I always think about the side effects. Knowing that I have a tumor that could respond to a checkpoint inhibitor, I will take that risk and I will take that drug. But we know that not everybody is going to respond to that drug. So huge area of research, really trying to understand what disease do you have, what is the best drug. And for those patients who currently don't do well with the treatments that we have, to really understand their disease and think about how to find a regimen that's going to help that patient. And to me, biomarkers are key to deliver on that promise. So we've been talking about biomarkers, and what Precision brings to the table here is really to integrate the clinical trial and the clinical trial design, all the work that goes into finding the right clinical sites and the best key opinion leaders to drive that study. But I'm very passionate about the labs and understanding what's going on with the patients once you get the drug on board. And being able to bring that together was something that I was very passionate about, and why I joined Precision to head the research team. Because to me, there's so much to think about to run a good biomarker program. And I love to talk about, I like to talk with our clients about their various different drugs. But too often I feel like the biomarker piece is maybe lagging behind. I'm sure everybody, when they think about a clinical study, they have a stake in the ground. 1st of September, we're dosing the first patient. And I have great conversations. This is an amazing drug. Have you thought about the biomarkers? And it's like, yeah, we should have a biomarker program. I'm like, it's June, guys. You're going to dose the first patient in September. So we really built Precision to be able to engage our scientists early on when we're thinking about a clinical program, because there's a lot of pre-work that has to be done. From the most simple things that often get forgotten. How are you going to stabilize a sample? How are you going to get that blood from a clinical site in Australia to a lab that can run a very complex 18-color flow cytometry phenotyping assay? How are you going to make that happen logistically? So that takes planning. You want to be able to inform patient care. You might want to change a patient treatment based on a biomarker. You need to have that run at a very high regulatory standard in a CLIA lab, which is why we built our CLIA labs. So to me, being able to deliver that integrated program is why I joined Precision, and we built an integrated labs and clinical trial program.
So just a little bit of history here. The National Institutes of Health in Bethesda, Maryland built a clinical building called Building 10 several decades ago, with the idea that patients would be treated here and their lab research would be treated in the next hall over. And in a way, Precision for Medicine is doing exactly that now. We're not in the same building because in today's world that's not practical. but we have worked on ways to get the samples of the patients viably to expert labs so that they can be assessed. From a research standpoint, we often use the word biomarker because that's what the specific assay that we may be looking at, but we may be looking at 10 different immunologic markers, and we may be looking at other biomarkers that have to do with the cancer cell, for example. And so it's really a concept that has developed in clinical research to be intimately involved with the labs so that you can understand what's going on in your patient in a much more specific and precise way. And that's what really created precision for medicine.
So that's a really good question. What would you lose if you don't integrate a biomarker into a clinical development strategy? Clearly a lot of really important information. Cancer is a very complex disease. You're never going to cure everybody with one particular drug regimen, not unless you're incredibly stratified to a very consistent type of cancer. And even then, I think you would be very challenged. So to me, you lose all the information about why a particular patient responded versus why did another patient not respond. And I know I've worked in this business a long time. Clinical trials fail. And should you get to the point where you don't make your clinical endpoint, if you've designed a good biomarker program and you've got different assays that let you understand the PK/PD of your drug, let you understand the type of tumor, let you really think about, did I have the true response that I would predict with my mechanism of action? Should you miss your clinical endpoint, you can go back and mine that data. The types of bioinformatic tools that Precision for Medicine bring to bear on this would help you really understand, did a subset of my patients respond? And maybe you can do a post hoc analysis and rescue that clinical study. So to me, to not have a biomarker program, you've really only got half a clinical study.
In the past, we didn't have these types of tools to really help us understand physiologically what's going on inside of our patients. Today we have many, many tools, and we're learning every day. In fact, we're learning from every patient that we treat how to make this better. And the rapidity of our progress is actually dramatic. Keeping up with that is difficult. And so the communication around which biomarkers are important is really an essential part between physicians, clinical trials, researchers, and patients and their families. Let me give you an example. Today, one of the hot kind of topics is whether a tumor is called hot or tumor is cold. But it's actually a continuum. There are warm tumors, really warm tumors, warmish cold tumors. These all react much differently than from each other in how the immune system is going to interact with the cancer cells themselves. And so cancer therapies that are immune-based have to consider this new data about hot tumors versus cold tumors. And one of the interesting things is we've discovered just recently that the checkpoint inhibitors, which have been very good in getting responses and some long-term survivals when they were never possible before, but it looks like the data's coming out pretty clear that you have to have a really warm to a hot tumor for those to work. And so therefore, we've developed a biomarker strategy with Debs and her group to help measure the hotness and coldness of the tumors within the patients in the clinical trials. And at the end of the study, then you can look at this data, see who responded, who didn't. Were you able to get responses only in hot tumors, or were you able to get them in warm tumors? Or if possible, maybe even get them in cold tumors is something that we really want to know. And there are certainly new therapies coming out that are attempting to make cold tumors more warm to hot. And so measuring these biomarkers is frankly essential to being able to interpret the data at the end of the study.
So Jerry mentioned something really interesting, which is the concept of hot and cold tumors. And that really means, is your immune system seeing that tumor? Can it get in there and attack the tissue? And of course, I'm sure everybody listening knows about checkpoint inhibitors. And we're really trying to understand, why do only a subset of patients respond? And now we're doing interesting clinical studies at Precision with our partners, where we're bringing in different ways of activating the immune system, because it's great if the T cells can get into the tumor, but they need to be tumor specific. So there's lots of really novel mechanisms of action out there, whether you're thinking oncolytic viruses, you're thinking tumor vaccines, how you could put that together with a checkpoint inhibitor and really be able to turn the tumor from cold, immunologically cold, to hot and help cure that patient. But you can't really understand what's happening unless you've got good biomarkers. You need to be able to take a piece of tissue. You need to look at, did my T cells get into the tumor? When the T cells got in, were they activated? Well, that's a biomarker assay. So being able to have all those different technologies, being able to look at lots of different markers on T cells in a tumor, those are the types of assays that we routinely bring to clinical trials now. So I'd really like to talk about some of the things that precision medicine has done, our strategy in action, if you will. Obviously, I can't name clients or particular drugs because that would let everybody know who I'm talking about. But I'm thinking of a particular study, very innovative mechanism of action. They ran a phase 1 clinical study. They had very small numbers of patients. It's a phase 1. And they didn't see the clinical effect that they were looking for. So you can imagine how disappointed everybody is. They've got a great new antibody drug. They go into what they think, based on their animal models, should have given them a good result, and it really didn't. So they came to us, is there any way to rescue this drug? And they'd been very innovative in the way that they thought about it, and they'd banked down a lot of frozen peripheral blood mononuclear cells, and they had banked down various pieces of tissue. So that particular drug would modulate the immune system. So because we'd saved those peripheral blood samples, we could then run assays on the bank samples and say, did that drug really modulate the immune system? And what we found was, yes, it did, but not in the way that we had initially predicted. So this particular drug had changed the ratio of regulatory cells to activated cells, and being able to understand that and then look at the types of patients, we realized that there was a subset of patients that had responded to the drug. And we were really excited because it was a little bit surprising to our scientists that with so few patients, we could actually see a strong effect just in a handful of patients. And that allowed us to design assays that would select these particular patients prior to therapy, who would then be predicted to respond to the drug. We're now testing it. I think early data is very promising. So to me, it was really exciting as a scientist to see that theoretical concept of biomarkers and bioinformatics come in and rescue what was basically going to end up a dead drug. And you see that a lot in the pharmaceutical industry, that you put a lot of money into a clinical trial, and it's almost like you get one shot on goal. And if it doesn't work, you've probably let a really important drug just might die. So having biomarkers can really help you rescue something like that.
Debs and I work together, as you can tell, a lot. And that's because we both feel very strongly that understanding the biomarker situation, which is a measurement of the systems that are happening in our patients, and how effective a drug is in a pathway, in cellular type therapies. These are really important. One of the things that we have noted is that we think so much about the application of the science that's being developed for the clinics, that we focus on that to help our clients get their product to market. And as Debs has mentioned and I've mentioned, the population that's being treated is really important. And so we work together before the first patient may even be treated to define how we're going to understand what populations this is working in. and we develop assays. We develop, it could be in our labs anywhere around the world, or all of them working together with samples to help define the patient population that we're actually going to treat. And then we specifically develop the clinical trial to try to get a higher response rate in those selected populations. In a way, what we're trying to do is be very particular about the patients that get treated, that they have a much higher chance of response and hopefully long-term survival. And that's what precision medicine is all about, being precise in the treatment of the patient. And so between our labs and our clinical expertise, we try to understand those populations, define them very carefully, and then get the clinical trial done with as few patients as possible that can demonstrate whether this strategy works or not. And I'm really happy to say that regulatory agencies from the FDA and others around the world also respond very positively to this strategy. They're very interested in getting drugs approved, even if it's for a small population that it may work in, but may work extremely well in that population. And so it's really been very positive for our clients to work together to design these clinical trials, the assays, defining the populations, the doses, the schedules, and it's been very successful so far.
So I think about how I would like to see the field progress in the next 5 years, and there's so many innovative things being tried right now. I see more and more biomarkers being used in clinical trials. You seldom see a clinical protocol now where people aren't thinking about biomarkers, and I'm really fascinated about how you might want to think about different areas of immunology and what that is bringing to the field. There's been some really interesting reports in recent years about how the microbiome, the diversity of the bacteria that live in and within your body, in your gut, how that affects your response to drug. That to me is truly fascinating, that as scientists, we really don't understand how we interact with our environment. For a long time, we just thought, well, they live in our gut and they help us digest our food. But it is so much more complex than that. Everything from allergy to response to checkpoint inhibitors. So I'm really fascinated to see how about the study of the microbiome is really going to change how we think about clinical studies in the future. I think cancer in some ways, your lifestyle affects your risk of cancer. It affects how you respond to drugs. And I think to bring together lifestyle and lifestyle intervention, which you could think about modifying the microbiome is effectively a lifestyle intervention, how that might change your response to a drug that modulates your immune system and lets you then deal with the cancer that's within your body. I think that's going to be a very rich area in the future for research.
I think treatment of cancer is only going to become more and more complex. I think the era of having one drug really make a major difference is waning, and I think that the combinations of many drugs is really going to be an important part of the next 5 years. You can see that already in many clinical trials where checkpoint inhibitors, for example, were used alone, but they only resulted in 15% or so long-term survival, and the goal is to have many more survive than that. And so hitting different precise locations in these cancer cells with several different drugs is really going to become an important part. In that, let me just back up just a little bit in that there are two systems that really have to be treated in a human that has cancer. One is the cancer cells themselves. They're clearly abnormal. Their genetics are abnormal, sometimes massively abnormal. They have survival mechanisms that allow them to grow and to continue to grow forever, basically. And that includes not only the mechanisms to continue to grow, but it includes the nutrition to grow, because you can't continue to grow without elements to make new cells. And it also means that they have developed systems, those cancer cells in those mutations, to actually hijack normal cells to produce elements for the cancer cells, like nutrients for the cancer cells, or parts of the cell walls, or other parts of the cell. But then there's a whole other aspect, and that's the immune system. And what we see in the last few years is really a focus either on the cancer cell or on the immune system and trying to modulate those. But what really I think we're going to see in the future is modulation of both of these at the same time. Drugs treating the cancer cells, drugs treating the immune cells, to try to really enhance the ability to kill these cancer cells. And hopefully we can start using the word cure again sometime in the not too distant future.
So it's interesting to me to think about what's going to happen in the world of oncology research in the next 5 years or so. I really would like to see personalized medicine making a true impact. It already is in lots of particular areas, but as a patient, to be able to go to your oncologist and to be told exactly what type of disease you have, and to know based on that molecular screening or based on that tissue typing, to be able to design a very specific regimen for that patient and know that you have a very high percentage of success. I think in the next 5 years, we're going to get better and better at being able to stratify patients onto the correct combination of drugs. I mean, Jerry raised a very good point earlier that the idea of one drug is going to cure your cancer is really not very likely. And when you think about many different drugs at times, they're going to interact with each other. So understanding that process, I think in 5 years, we're going to get much, much better at being able to do that.